We started talking about the sample long FRQ, but just for the purposes of, we're very short on time, so we're going to just summarize what's going on in this paragraph, this figure 1, and then figure 2, and then table 1, which is on the next slide. Okay, so in this sample long FRQ, in this first paragraph, it's going over a species of mosquitoes that are responsible for transmitting malaria from people to people. And, one way to control the mosquito growth is by using an insecticide, okay? And the specific insecticide that is addressed in this FRQ are called pyrethroids. I think that's how you say it. Pyrethroids, okay? And these pyrethroids, they're going to target this transmembrane protein, okay? So this is what we call a transmembrane protein. A transmembrane protein is a protein that goes through the plasma membrane, and it looks like to me that it's in tertiary structure. This is a polypeptide chain, but it's in secondary, which is not shown. But then it's folded even further on itself, which means that it's in tertiary structure. And it goes through the membrane in 4 different places. Okay, that's why you have segment 1, 2, 3, 4. But it is one long polypeptide chain. And so the pyrethroids, they target this protein. And it ends up killing the mosquito. All right? However, in one common mutation to the channel protein, a phenylalanine amino acid is substituted for leucine at the amino acid position 1014. So along this polypeptide chain, on position number 1014, instead of a leucine, it's now a phenylalanine. So this is a mutation, right? And scientists hypothesize that because of this one mutation, it's responsible for pyrethroid resistance. So remember, pyrethroid is an insecticide, and more it's becoming widespread, this resistance. Oh, somebody just entered and their name popped up. I hope it's okay. I wonder if I could just get permission from that person to let their name just pop up. What do you guys think? Okay, well, we'll deal with it later. You know what? Maybe in when I import this video into iMovie, I can just delete that little part. I think I can do that. Okay. And in fact, I think I can import like a little shape, and maybe I can black out. So this is figure 1, which shows this transmembrane protein in the plasma membrane of mosquito cells. Okay? All right, to investigate this, insecticide resistance, mosquitoes were collected 4 times over a 2-year period from the following 2 regions. Region A. So region A is an area where insecticide use there's a large amount of insecticide use, whereas region B is an area where there's very little insecticide use. Okay? Yes, I'm using QuickTime to record. Is that... I don't have any other screen recording tools. So scientists, yes, there is. And I could lower the chat box, and I could make... that is a really good idea. I could have just lowered it, but then it crowds everything together. I think I can edit it in iMovie. So what scientists did was they collected these mosquitoes, and they used filter paper. And they used filter paper that was soaked in these insecticides. Okay, and there's actually 2 different kinds of insecticides in the group called pyrethroids. They are the deltamethrin and the permethrin. So those are the 2 different kinds of insecticides that are called the pyrethroid insecticides. And then after these mosquitoes were exposed to these 2 different kinds of insecticides, their mortality, percent mortality was collected. So percent mortality, remember, mortality means death. Okay, it means death. So it was recorded. So this is region A, where they were exposed to a lot of insecticide. Region B, where they were not exposed to a lot of insecticide, but then these these collected mosquitoes, they were exposed to either deltamethrin or permethrin. Okay? So this is figure 2, susceptibility of A. gambia mosquitoes from 2 regions to the pyrethroid, deltamethrin, and permethrin. A mosquito strain that is susceptible to the insecticides displayed at least 95% mortality in all experiments. So in the experiment, the scientists included a strain of mosquitoes that was known to die 95%, at least 95% mortality when exposed to the insecticides, okay? And then also, you guys, take a look here. Mosquitoes were exposed to untreated filter paper. So the filter paper did not have any insecticide. It was probably just like water or something, okay? And they displayed less than 10% mortality. And then here, in figure 2, you can see these bars, right? These bars, those are called the error bars, represent standard deviation, and ND means no data are available. So in region A for October 2008, there was no data available. Also, I mentioned in class that notice these graphs. These graphs are bar graphs, but it's on its side. Okay, it's on its side. So what is the independent variable? When you guys look at these two graphs, what's the independent variable? Can somebody let us know in the chat box? What's the independent? Yeah, the independent variable in this case, it's these graphs are flipped, okay, on its side. So it's actually the dates. That's okay. It's actually the dates. That would be the independent variable. You know, remember with experiments, whatever is being measured by the scientists, that is the dependent variable. Dependent. And what's being measured? Percent mortality. Okay, so that would be the dependent variable. And you know how in class they said, oh, you guys, the independent variable is always on the x-axis, and then the dependent variable is always on the y. Well, in this case, they flipped it on its side. Okay? So, yes, I'm sure you know, you know that, you know it. Okay, and then here's table 1. I know, you know what, they want you to be able to understand what is the dependent variable, what is the independent variable in an experiment. Dependent variable is always what's being measured. Somebody says, wait, independent x was what? The independent variable in this case, so going back to the slide, the independent variable was time, okay? The dates that the mosquitoes were gathered, okay, collected, the months and the year, okay? All right, here's table 1, and table 1, they studied the amino acid sequence of that transmembrane protein, and in this table you can see there's the region A mosquitoes and then the region B mosquitoes, and those are the 4 dates where they were gathered, and they were studied, some of them, you know, were studied for homozygous for leucine, heterozygous for leucine and phenylalanine, or homozygous for phenylalanine. Okay, so you have region A and region B. Okay, let's take a look at the question. So when you guys turn in your Google slide document, you need to write your answers in this yellow box, okay? In this yellow, I want you to type in your answer. Let me just see here. Why is, Creeper Allman says, why is homozygous for leucine missing 3 slots? Okay, so when they gathered the mosquitoes in region A, none. Zero mosquitoes in June 2009 were homozygous for leucine. 0 mosquitoes that were gathered in October 2009 were homozygous for leucine. And then 0 mosquitoes out of the 46 that was collected in June 2010 in region A, 0 of them were homozygous for leucine. Majority of them were homozygous for phenylalanine. That's what it means, okay? Sure, no problem. How can you be homozygous for an amino acid? You take a look. Well, okay. We did talk about how you could do DNA let me let somebody in. And oh no, their name popped up again. Okay, how can you know? You can do DNA sequencing, and then you can do restriction enzymes that cut known DNA sequences, and you can figure out if you're homozygous, heterozygous that way. Or you could do protein analysis. You can actually do protein analysis. Okay, that's how you can find out if a protein, if an individual is homozygous for leucine, heterozygous, or homozygous for phenylalanine. Okay? All right, let's go ahead and talk about part A. Describe the most likely cause of the amino acid substitution in the sodium channel protein. Remember this, going back to the question here, it says in the introduction that there in some mutated mosquitoes, phenylalanine is substituted for leucine. How did that happen? Okay, and Witty from FNF. I'm not sure what that references, but right, what's mutated? Can you be a little bit more specific? What has mutated? Anybody? When we say mutation, what is that usually referring to? Yeah, because you guys remember when, remember DNA? It codes for an mRNA codon, right? And what do codons code for? Amino acids, okay? So you're gonna have first a mutation in the DNA, which is probably or can change the mRNA codon, which then can code for a different amino acid. So instead of leucine, now you have phenylalanine because of the DNA change. Phenylalanine? I forgot how to spell it. What do you call an acid with an attitude? Oh yes, I've heard that joke before. Amino acid. That's a good one. And now it'll go down in YouTube history. Okay, also part A. So part A is asking about, what's the likely cause of the amino acid substitution? Now for part A, second part of part A, because now it says, here's another verb task, explain. So the first one was describe. Now explain. Explain how the substitution of a single amino acid in the channel protein could cause pyrethroid resistance in mosquitoes. So going back to this picture, it says here that at amino acid location 1014, there's been a mutation. How come the mosquitoes with the mutation, how come they are resistant to the insecticide? So it's really asking, like, how come the insecticide, and it says insecticide. It says it right here that the, let's see, insecticide, right here, pyrethroids interfere with the function of a transmembrane sodium channel. Okay? All right, let me go ahead and read what's going on in the chat box. I do believe I have to. The codon is the change in the codon is what's going to cause a change in the amino acid, but it's really asking, like, how is it going to affect, the insecticide? Because that mutation will be passed down to future generations. That's true. That is an example of natural selection where the ones that are resistant to the insecticide, they're going to survive and reproduce. But it's really asking, the question is, explain how the substitution of a single amino acid in the channel protein could cause pyrethroid resistance, okay? And the next person, Creeper Allman, says it changed the structure and shape, which changed the function of the protein. You guys, the mutation has changed the shape of the protein, and now can the pyrethroid, which is insecticide, can it target this protein and interfere with its function? No, right? So kinase West, yeah, maybe they can't be inhibited anymore. Exactly, because there's a change in the shape of the protein. Exactly, Hazelnut. All these names are so funny. Change in the shape stops the binding of the insecticide. Exactly. So in this case, it's not a frameshift mutation, because the reason why it's not a frameshift mutation is because if you look here, it says that there's only one amino acid that's been substituted. If it was a frameshift mutation, excuse me, all of the rest of this polypeptide chain would be changed. Okay. All right. So no, it is not a frameshift mutation. It's probably a point mutation, and it's called a missense. Do you guys remember that word, missense? Missense point mutation, where it changes the amino acid. Okay. So that is part A, everyone. That's part A, where you're going to type in, mutation in the DNA, which changes the mRNA codon, which changes the amino acid, right? And then explain how the substitution of a single amino acid. So some of you guys put in some excellent answers there. Let's move on to B. Identify the dependent variable in the experiment whose data are graphed in figure 2. And we already kind of went over that, right? The dependent variable is the percent mortality. That is always what's being measured in an experiment. Whereas time, the groups, okay, and whichever insecticide they're receiving, June 2010, that's the independent variable, okay? And then it says right here, identify the positive control. Do you guys see that? Positive control. We haven't really talked about that. But a positive control in an experiment is part of an experiment where scientists, they expose, in this case, the mosquito to a known insecticide that is known to kill mosquitoes. They know the outcome of the treatment. So that's a positive control. They know the outcome of the treatment. It's not the same thing as a control, you guys, okay? It's not the same thing as a control of the experiment. This is positive control, where they expose the organism, in this case mosquito, to a chemical, and they know exactly what's going to happen. So if you go back to figure 2, it actually tells you very quickly, and you could definitely have missed this. This is the positive control. A mosquito strain that is susceptible to insecticides, meaning that they can be killed by the insecticide, displayed at least 95% mortality in all experiments. So they wanted to use a positive control. The whole purpose of a positive control is to make sure that everything is working in the experiment, that mosquitoes will die when they are exposed to insecticide. So they took a strain of mosquito which did not have this mutation, no mutation. They knew it was going to be dead, just to test to make sure that, these insecticides are actually working, okay? Somebody says, I'm confused, isn't the dependent variable on the y-axis? So wouldn't time be the dependent? Okay, you know what? I have said that to you guys in class. I have said, you guys, the independent variable is always on the x and the dependent variable is always on the y. But this is a case where they flipped it, okay? This is a case where now what they are measuring is on the y, and then what's being changed is the x-axis. So just remember that, for me, I literally imagine scientists measuring things, okay? They're measuring how many mosquitoes are dead, right? That's the dependent variable. Dependent variable is always, what are the scientists measuring? At the end of the experiment, what are they looking for? They're not looking for, it's June 2010. They're not measuring time. They're measuring percent mortality. Okay? So, yeah, I don't know why they did it this way, because honestly, couldn't they have easily just done it this way? They really could have, but they didn't. They really want you to look at the graph and figure out and understand, have a good understanding what a dependent variable is. Okay, so somebody asked me, can you tell me what the positive control is again? A positive control is part of an experiment where they know the outcome. They know the outcome. So this is the positive control right here. A mosquito strain that is susceptible to the insecticide. It displayed at least 95% mortality in all experiments. This is a group where they knew that the mosquitoes did not have a mutation. They knew that these mosquitoes would be killed by the insecticides, so that they knew that the insecticides and everything in the experiment is just working. Okay, so that's a positive control. It's like a type of treatment, but the scientists know exactly what's going to happen. Yes, so this would be the positive control. A mosquito strain that is susceptible to the insecticide. They knew this was going to happen, and it's part of the experiment. Gosh, there's so many. There's examples. Let me give you an example. Like, let's say that scientists want to study if a fertilizer is working, like a brand new invented fertilizer. They already know which fertilizers work. So what they'll do is they'll allow for plants to get that fertilizer that they already know that works. That's the positive control, because they already know what's going to happen. And then they use that to compare the new fertilizer. And then they also have a control group without the fertilizer. Does that make sense? So in that specific example I'm giving you, it's like 3 different groups: a control, a positive control, and then the actual testing of the fertilizer. Somebody asked me, is it like a confirmed outcome? Yeah. Like we know that we know the insecticide works, and we're just going to be comparing how, these mutated mosquitoes, how do they withstand? What's their mortality rate? Okay, so going back to this slide, so identify the positive control, we just said that. Next question, justify exposing some mosquitoes to untreated filter paper each time the experiment was performed. So these filter papers, they do not have any insecticide. They probably just have water. What is this called, by the way? This group of mosquitoes that are exposed to untreated filter paper in an experiment, what group of mosquitoes? What group do they belong to? Exactly, the control group. No, positive control is different. Okay, positive control is different. Positive control is the non-mutated mosquitoes. They're exposed to insecticide. We know they're going to die. There was a 95% mortality rate. Okay, at least a 95% mortality. And then for the control group, there was less than 10%. So, you guys, basically, these are like these are the experimental groups, the mosquitoes from region A and B, and then you have a control group, and then you have a positive control group. Okay, so really, this experiment has like at least 4 different groups, okay? Because the experimental group, which has region A mosquitoes, region B mosquitoes, and then you have a control group. They're exposed to untreated filter paper. It was probably just water on filter paper. Positive control, these are the mosquitoes that do not have the mutation, and they're exposed to the insecticide. We know they're gonna be killed. Okay? Okay, and then actually somebody asked me a good question. What is the control used for? What's its purpose? So this is the control group. What is the purpose of a control group? And that's part of justification, you guys. Justify exposing some mosquitoes in untreated. Why do we do that? Well, it's called the control, so you have to use a different word. Yes, control serves as something to compare to. Exactly. Yeah, it could be, you can say baseline. The mosquitoes that are just given water and no insecticide, a standard, exactly. They're gonna live, right? They had less than 10% mortality rate. So we can use that to compare positive control and then also these region A mosquitoes and region B. Yep, exactly. Okay. Somebody says, so the justify would be that the control, yes, so the justify, so this part of the question, justify is, you would say that the control group, it is the control group, so the untreated filter paper mosquitoes, they are in the control group, and it's going to be used for a comparison or a baseline, as you guys said. to study. You can even go into detail, like, to study how insecticides, their effects on the population. Okay, so here we have part C and D. Part C, and notice how part C, guys, there's like 3 action verbs. No, positive control is not a control group. It is not the control group. A control group usually gets no treatment, okay? A control group gets no treatment, so I deleted it, but the control is the group that only gets like water on the filter paper, and then the positive control, they are the mosquitoes without the mutation. They get the insecticide. We know their mortality rate is gonna be high. We know they're gonna be killed. Okay, so it is not the same thing. Somebody says, so positive is like, we know the results. Control is like, use as comparison. Very good way of putting that. Okay, so let me repeat what ... said. So positive is like, we know the results. Control is like, use as a comparison. Yeah, both are going to be used as comparison though. Okay. So based on the data in figure 2, describe whether mosquitoes from region A or from region B are more likely to exhibit greater evolutionary fitness. You guys know now that evolutionary fitness means that they're able to survive and reproduce. Okay? So which group, A or B, which one is better able to survive and reproduce? So let's take a look at figure 2, because it's saying based on figure 2. Okay, so here's figure 2. What do you guys think? Which group, region A or region B? Oh, interesting. There's different answers in the chat box. So if you take a look at the graph, percent mortality is the amount of deaths, right? And when I compare region A with region B, which group of mosquitoes has a higher mortality rate? Okay, I know Witty is just joking around. So B has a higher mortality rate. So which one has a higher fitness? Which one has a higher survival rate? A. Okay, do you guys see that? Especially the ones that are exposed to this specific pyrethroid insecticide, permethrin, they have a very low mortality rate. It's only like 20% or less, okay? 20% or less are going to actually die, 80% are going to live. So there's a higher survival rate amongst mosquitoes in region A, okay? All right, so describe, let's move down here. Describe, so based on the data in figure 2, describe any significant, you guys, this word significant, remember we talked about that when it comes to those error bars, okay? Describe any significant change in the susceptibility of mosquitoes from region B, just region B, to each of the 2 insecticides over the 2-year period, okay? So we're gonna look at just mosquitoes in region B. Speaking of mosquitoes, there's a mosquito flying around my head. Okay, so just look at region B. We're just looking over here. So you guys, let's look at October 2008 to June 2000. Was there a significant difference? What do you guys think? Between 2008 and 9? What do you guys think? Yeah, not really, but it could be. I mean, there could, maybe there's no overlap. So if there's no overlap, then we do say there is a significant increase in mortality, right? There's an increase in mortality from 2008 to 2009. But what about from June 2009 to October? Would you guys say that's a significant decrease in mortality? Do those bars, do they overlap? You know what? There's no overlap, so we're going to say, oh gosh, that's supposed to say overlap. There's no overlap, so we're going to say there is a significant difference. There is a significant difference. It is kind of hard to tell, but there's no overlap, so there is a significant difference in mortality rate between June 2009 and October 2009. What about 2009 to 2010 in this group? Is there a significant difference? It's really hard to tell, huh? But is there, what do you guys think? Is there overlap between the bars? There's no overlap, so I would say, oh, sorry. Yeah, there's no overlap. So there is a significant difference. So no overlap between those two dates as well. So I would say that there is significant difference between June and October, and then between October to June 2010. So there's no overlap. So that means there is significant difference. Okay? You guys, so that means there is significant difference because there's no overlap. Let's take a look at the group that got the permethrin. Is there a difference between October 2008 and June 2009? What do you guys think? I feel like there is overlap, huh? So there's no significant difference. What about between June 2009 and October 2009? There's definitely overlap, so there's no significant difference. But what about between October 2009 and June 2010? Do you guys think there is a significant difference? So you guys, for the next part of the free response, that's basically what you're saying. Is you're going to say that for region B, the mosquitoes that were exposed to permethrin did not see a significant difference between October 2008 to 2009. However, there was a significant difference from October 2009 to June 2010. So right now I'm telling you guys what you need to write, okay? But for the mosquitoes that were exposed to deltamethrin, there was no significant difference between October 2008 and June 2009. But there was a significant difference between June 2009 and October 2009. And then there was a significant difference between October 2009 and June 2010. Okay? So you guys, for this question, basically what I'm doing is I'm just comparing the bars from month to month or year to year. That's all I'm doing. Okay? So the group in region B that was that got the permethrin insecticide, there was no significant difference in mortality rate from June 2008 to October 2009. There was, however, when I compare these two bars between 2010 and 2009, October, there is a significant difference. So the mortality rate went down significantly. Okay? Mortality went down significantly from October 2009 and June 2010. And then for this group up here that got the deltamethrin, there was no significant difference between October 2008 and 2009. However, there was a significant decrease in mortality from June 2009 and October 2009, and then again from October 2009 to June 2010. Sure, go ahead, Witty. Just to be sure, mortality is higher death rate. Mortality is just death. So percent death, that's what this is showing. So like, for example, October 2008, for this group, there was a death rate between, I would say, like 75 to 90% of them died, like that. Okay, is Woody going to recap or? Okay. Oh, I was asking if I was right. Where did you recap? Wait, let me recap to see if I remember if overlap. Oh, I see. Yeah. Yeah, overlap, no difference. If no overlap, yes, difference. Exactly. Okay? All right, so that's the answer for C. Oops, a daisy. Oops, I moved it by accident. For C, where it says describe. Okay, yeah, basically C2, exactly. Okay, and then next, you guys, this is going to be sort of like Hardy-Weinberg, everyone. Calculate the frequency of the allele coding for phenylalanine in each population of mosquitoes in October 2008. Frequency of phenylalanine in each population in October 2008. Okay? And it says here, round your answers to 2 decimal places. Okay? I know some of you guys are saying PTSD, right? All right, so we're only looking, oh wait, hold on, I already forgot. Is it B? Oh no, it's both population. Oh wait, let's see, in each population, October 2008, okay. All right, so we're looking at phenylalanine only. Okay, phenylalanine only in the 2, oops, let me undo that because it's not the entire populations, it's just October 2008. So we really only want to look at October 2008, October 2008 for A and B because it says the 2 populations. Okay, so the guys, this is how you do it, right? You first, it says right here, total mosquitoes tested 39. So how many alleles total is that? Okay, so let's do this A. So if it's 39 mosquitoes, how many total alleles are we dealing with? Good. My name said 39 times 2, which is 78, okay? 78. So now let's go ahead and count how many phenylalanine alleles there are. This one is heterozygous for leucine and phenylalanine. That would be 5, so 5 plus. And then homozygous for phenylalanine. There's 31 mosquitoes that are homozygous for phenylalanine. That means that they have 2 each, so you need to multiply that by 2, right? And that gives you 62. So when you guys add that, that's 67 divided by 78. That's going to give us the allele frequency for group A in October 2008. So let me go ahead and divide 78. And that gives me 0.86. 0.86. Okay? Okay, and then for B, okay, for B, there's 27 mosquitoes. So you guys, what's the total number of alleles for 27 mosquitoes? Remember, it's 27 times 2. That gives me 54 mosquito alleles that we're dealing with. And this is 5 plus, and there's only 2 mosquitoes that are homozygous for phenylalanine. So you multiply that by 2, right? And that gives us 4. So that's 9 divided by 54. And that gives me 0.16. Or 0.17 actually. Let me actually round that to 0.17. Okay, there is a question in the chat box, which I'm going to read in a sec. Is it specifically asking for the ones in October? Yeah, if you guys go back to the question, it says here, October 2008. Calculate the frequency of the allele coding for phenylalanine in each population, that's A and B, for October 2008. So we were only looking at October 2008. Okay? So everybody got that? Alrighty, and then let's move on to the next question, D. Oh, somebody said, wait, no. You know what? I already deleted the answer, but it was, I mean, yeah, we can look at the recorded, and then I believe it was 0.8, what was it? 0.86 and then 0.17. Okay. Okay, let's go. Oh, you guys, look at the time. And it's okay. We can stay for as long as you guys want. Okay? All right, for part D, using mosquitoes from insecticide-free areas, the scientists developed mosquito strains with amino acid substitution at other positions in the sodium channel protein. They exposed the mosquito strains to non-pyrethroid insecticides. Okay, you guys, I'll be honest with you here. Okay, let me just finish reading the question and then I'll tell you my problem, okay? Predict the susceptibility of the mosquitoes to the insecticide. So we're talking about non-pyrethroid insecticides. And what the scientists did was they created these mosquito strains with other mutations on that sodium channel protein. Predict the susceptibility of the mosquitoes to the insecticides. Susceptibility means like how they are affected. Are they going to die? Are they going to live? How susceptible are they to the insecticide? So my thing was like, first when I first read this question, I completely ignored the non-pyrethroid insecticide. So these are insecticides that do not target that protein. Okay, so do you guys understand? They are now, what they did was they created mutated mosquitoes with mutations on that protein, but then they exposed those mosquitoes to insecticides that were not non-pyrethroid insecticides. So what do you guys think? Do you think the mosquitoes might die from these insecticides, or do you think they're going to survive these insecticides? What do you guys think? Don't worry, if you get it wrong, it's okay. Okay, so you guys are saying survive. All right, if you look at the answer, and by the way, the help button will take you to the answer. It will be on YouTube, yeah. Actually, the mosquitoes will die, okay? They're gonna die. Because these insecticides, they do not target the sodium channel protein. So even though the scientists, they mutated the sodium channel protein, that's not the protein that the non-pyrethroid insecticides even target. These are other insecticides that probably target something else, okay? So the answer for this one, predict they're gonna die. The mosquitoes will die when exposed to these insecticides, okay? Because again, the non-pyrethroid insecticides, they're probably targeting something else. It's the pyrethroid insecticides that target that sodium channel protein, but we're using something else. We're using a different insecticide, okay? Okay, last thing. The scientists claim that the mosquito population of... Can you explain that again? Okay, so remember this insecticide, the pyrethroid insecticides? Remember, those insecticides, they target the sodium channel protein, right? But in this example right here, they're not using pyrethroid insecticides. They're using something else. So these insecticides are going to target the mosquito, and the question is, predict what's going to happen to the mosquitoes. Well, they're going to die. The mosquitoes are going to die because we're talking about a different type of insecticide, okay? It has nothing to do with that sodium channel protein. Okay, I know, I feel like this part of the question was not very clear. It's only worth 1 point. Predict the susceptibility, oh, I already went over that. The scientists claim that the mosquito population of region B evolved resistance over the period of the experiment, and that resistance arose as a result of immigration of resistant mosquitoes from other regions. Based on the data in Table 1 and the information provided, provide evidence to support the scientists' claim. So, you guys, what they're saying is that the mosquitoes in region B, the reason why they became resistant to the insecticide, the pyrethroid insecticide, is because of immigration. Mosquitoes that were already resistant to this insecticide, they moved in to region B. And it says here to go back to Table 1. Yeah, exactly, gene flow. Exactly, gene flow. So what they want you to do is go back to Table 1. Remember, these guys are the ones that we're talking about. And remember, the mosquitoes from region B, they come from an area where there's low insecticide use. Remember, they come from a low insecticide use area, whereas region A, they come from an area where there's a high insecticide use. Okay? And so you guys can see here that, remember, the phenylalanine mutation, though phenylalanine causes them to become immune or resistant. They become resistant to the insecticide, the pyrethroid insecticide. Okay, so you guys can see here that over time, from October to June, and then from October to June 2010, there's an increase in the number of resistant mosquitoes. There's an increase. Okay, there's an increase. And the scientists are saying, oh, this is due to immigration, not due to them growing in resistance. Okay, so the question is, it's saying use evidence from this table. Let me just go back here. Provide evidence to support the claim that it's immigration and not a result of evolved resistance. Okay, so the scientists are saying it's immigration, not evolved resistance. The key here, you guys, I don't know if you guys know the answer, but the key here is that region B, they were not exposed to a lot of insecticide. So why would they become resistant to the insecticide if there's not a whole lot of insecticide being used? Does that make sense? Okay, so region B, there's not a whole lot of insecticide being used, so the environment is not going to choose mosquitoes that are resistant to insecticide because there's not a lot of insecticide being used. Does that make sense? So why is the population changing from non-resistant to resistant? It's because of immigration. He says, couldn't they have developed a mutation, or that not likely in such a short amount of time? No, it's possible. It is possible to get a mutation. But then again, these mutations, here's the thing, there's a huge change in gene frequency. It's going from like 2 individuals to 12 that are homozygous. That's a huge change. And so if it was beneficial to be resistant to the insecticide, then the ones that are resistant will survive and reproduce. But there's no insecticide being used in region B, so there's really, they're not being selected for. So the scientists are saying it's due to immigration. Okay? All right, so you guys, that was the very first FRQ. A lot of images. Okay, and then by the way, you guys can click on here and go to the answer. All right, let's move on to the short example, okay? The short FRQ. And like I said, these short ones are only worth 4 points. This is a sample question number 5. And sample question number 5, according to the College Board, they say that you're going to be given a model or a picture that you need to interpret using the content knowledge that you have learned. Yes, they do. By the way, last year, what they did last year was they actually broke it down. So part C was actually broken down into 3 parts. So last year it didn't go up to D, part D. It actually went up to G and J, all the way up to, one question had up to J. It was A, B, C, D, E, F, D, H, I, J. Literally, they had, because they decided to break it down for our students. See these action verbs, describe, calculate. They made it into separate parts so that students wouldn't get confused. I don't know. I feel like it's better. I think it's better if they because some students might skip parts, right? But then they broke it down into several parts. Okay. This year, I don't think they plan on doing that. I think this year they plan on keeping it like this, where part C has 3 action verbs. Part D has 2 action verbs. Somebody says, I wish there was a help button on the AP exam. Yeah. Okay, so you guys, let's take a look at this one. This is the last one. In humans, the gene that determines a particular condition has only 2 alleles, one of which, big B, is completely dominant to the other, little b. The phenotypes of 3 generations of a family, and this is a pedigree, right, with respect to the condition are shown in the pedigree in figure 1. Individuals are numbered, Okay? So part A, describe the process in which eukaryotes that ensure that the number of chromosomes will not double from parent to offspring when gametes fuse during fertilization. So you guys, part A has nothing to do with this pedigree. They want to ask you, how do eukaryotes, how do they make sure that at fertilization the zygote does not have double the number of chromosomes? Do you guys remember? What's this process that makes sure that when eukaryotes, reproduce, they're not doubling the amount of gametes or chromosomes? Meiosis, okay? Somebody mentioned checkpoints. That's a good point. But in this case, we're just talking in general. It's meiosis, okay? Meiosis. We're just talking about meiosis in general. Because you guys remember in meiosis, it's a process where the number of chromosomes are halved, right? So we're going from 46 chromosomes to 23, right? And then the homologous pairs are separated. Remember? The homologous pairs are separated. So now we have just one set. We're going from 2N, right? We're going from 2N diploid to haploid. Now each cell has just half the number of chromosomes. And then the sister chromatids will separate. So now you have the sister chromatids separating. And it's still haploid gametes. Okay, 1N, haploid gametes. So the answer is meiosis, okay? The separation of homologous pairs in meiosis. All right, let's do part B. Explain how any one chromosome in individual 16, right over here, contains DNA that came from both individuals 1 and 2. How is it possible that person number 16 got DNA from both of these, I guess, grandparents, right? Good, my name, crossing over. Remember in meiosis, prophase 1, the chromosomes cross over, and then you're going to end up with, chromatids with pieces of maternal and paternal, all right? So eventually, those chromosomes are going to be passed down from parent to offspring. Yes, crossing over. They do hug, okay? So that's what we're talking about. So he says something important in the chat box. The wording of these questions are making me lose my mind. You guys, this is why practice is so important. Because once you get, trust me, you are not the only one to feel this way. Once you get to see like at least 10 to 20 of these type of questions, you're like, okay, that's, I'm used to this now, and I can do it, okay? You're losing your mind now, but the more exposure you get, the more practice you get, you're gonna be able to do it very easily. Okay, actually part C, it does have to do with the pedigree, okay? So let's go ahead and answer. Let's go to part C. Use the template, and obviously you're not going to get a template because yours is all digital, okay? Use the template figure of the pedigree and the allele designations big B and little b to indicate the genotypes of individuals 2, 4, 8, and 18. Okay, so individual 2, they want the genotype. Individual 4, what is the genotype? Individual 8, and then individual 18. Wait, why does it remind you of Michael Jackson? That's kind of weird. Oh, he. Oh, okay. So you guys remember at the beginning they said big B or little b, right? And with the condition, they're filled in. So you guys, what are we talking about? Are we talking about, oh, you know what, down here for part D, explain whether the inheritance pattern of the condition is sex-linked or autosomal and dominant or recessive. You guys, here's the thing. They already told you whether or not it's dominant or recessive, didn't they? Oh, but you know what? They're asking about filled in. Is the filled in individuals showing the dominant or the recessive trait? Okay, that's what they're asking. So let's try to do this. Well, let's try to figure that out first. What do you guys think? Do you think it's recessive or is the filled in conditions, is it showing dominance or recessiveness? Recessive, okay? The reason why, you guys, the reason why it's recessive is take a look right over here. This individual 14 has it, but the parents do not. The parents, if it was dominant, then more than one kid would have it. It's impossible for one kid to have it and none of the parents to, okay? So what we're talking about is recessive, okay? So this is little b. That means that the parents are probably heterozygous, showing a normal, dominant trait, but then their little kid has it. So then, therefore, number 1 is little b. What would be the genotype of this person number 2? Anybody know? Let us know in the chat. Yeah, they're heterozygous because they have a kid that's little b. The only way to get a little b kid is if this parent is heterozygous. Okay, and then it says person 4, right? Oh, we already decided what person 4 is, big B, little b. And then person 8 is little b. And then what is person, you guys, what is 18? What do you guys think is 18? Person 18. Okay, big B, little b. How do we know that? Well, if you take a look at, these parents, they have 3 kids, right? They have 3 kids. One of their kids is little b. So this kid would have gotten the little b from the mom, and then they must have gotten that little b from the dad. And the dad doesn't show the trait, so it's got to be, the dad must be big B, little b. And then this number 18 must also be big B, little b. Okay? So we did figure out the genotypes of the individuals based on, okay, what do you guys think? We know that it's recessive, but is it sex-linked or autosomal? What do you guys think? Is it sex-linked or autosomal? Yes, 2 would be big B, little b. Yes, this is autosomal. Okay, this is autosomal. The reason why that it's autosomal, okay, and the answer key says something like if it was Y-linked, then the males would have it, right? So like if you look over here, if it was Y-linked, this dad has it, then his son would have it, but it doesn't. But then if you take a look at, let me just find a female. Oh, right over here. Let me go ahead and show, I'm gonna have to clear this, okay? If you take a look at this female right here, this female has the condition. If it was sex-linked, then she would be like X little b, right? And if she has X little b and X little b, then her sons will get it as well, right? Because the sons are going to get that X little b from the mom and then the Y from the dad. So this is why it's autosomal, is because when you look at these 2 parents, the mom has a condition, then all of her sons would, okay? And then also somebody mentioned that, oh, you guys all mentioned in the chat box that there's more than one set, well, there's equal number of males and females, that there's similar numbers, okay? Okay, and then let's see, I think, so the rest of the slides are basically the answers. The help buttons will take you to the slides with the answers. Somebody asked, for part C, are we required to explain why each individual is what genotype? Let's go ahead and see what they have on the rubric. That's a very good question. It says here, indicate the genotypes. It just says indicate. Okay, that's the action verb. The action verb is indicate. You just have to give the genotype. But if they say indicate and explain, then you would need to say why. Okay? So if they say indicate and explain, or if they say justify your reasoning, okay? Then you would have to give a reason why, and maybe you would even have to show a Punnett square. Like if this parent was like big B, little b, then you would have to do a Punnett square and all of that stuff, okay? Okay, thank you, thank you. I know who you are if you shared the slides. So you guys, you get 10 quiz points for doing this Google slide document. I do want you to type in your answers in these yellow boxes. I don't want you, well, we went over the answers together, but I want you to type it in how you would have answered it, and then I want you to compare your answers to the rubric. Okay? So you're going to be getting 10 easy quiz points. Okay? But the whole point of doing this, to make it meaningful, right? We want to make our time meaningful. We want to make the review for the AP exam meaningful. And the only way it's going to be meaningful is for you to try to answer these questions on your own. Like this guy was a really long one. Try to answer it on your own and then compare it to the rubric. And then see where you could have improved, where you can improve. Oh yeah, it'll definitely improve your grade because you're getting 100% on a quiz. You're getting 10 out of 10. We're doing FRQs for the next 3 Mondays. Oh, actually there's 3 Mondays left. There is a Monday where there's a day off, but yes. And you will be getting 10 easy points every time we do this, okay? Creeper, oh, sorry, it's a direct message, but you guys will see it in the chat. In my shared screen. Somebody asked me, I was curious, are you still giving a final even though... No, I am not giving a final. Wait, are we going to get 6 FRQs on the AP exam? Yes! Yes, remember we went over that today at the beginning of class? 60 multiple choice, 6 FRQs. The first 2 FRQs are going to be long. The last 4 FRQs are going to be worth 4 points each, okay? So if you take a look at this document, you will see exactly what type of questions each FRQ is going to be, okay? Yeah, there is, I think the 31st of May is Memorial Day. We have the day off, okay? You guys, I challenge you to try to answer the questions on your own and then take a look at the rubric. You will not get marked off, okay? I have a question about tomorrow. On our quiz, we have 15 yeah, tomorrow's quiz is all multiple choice, 20. It's 20 multiple choice questions on Unit 2, okay? When it comes down to FRQs, my world history teacher tells us to divide time so we didn't spend too much time on one and rush through the next. How can we divide time with these types of FRQs? Okay, so what I would recommend, and also the College Board has said, the first 2 questions are always the longest. You should spend about 20 minutes each. Like 20, they say like, you know, about 20 to 22 minutes on each of them. So that would give you like 44 minutes to do, you know, that's like 45 minutes to do the first 2. And then the last 30 minutes would be divided amongst the last 4 questions. Oh, wait, that doesn't add up. I'm sorry. 45, 45, okay? So 45, 45. So split it up that way. 45 and 4, because you have 90 minutes total. So then that means that for the short ones, you get like 10 to 12 minutes or so, okay? All right, and then, let's see here. Witty says, I challenge the viewers to smash that like and subscribe. How much time do we have? So again, multiple choice is 90 minutes, and then free response is also 90 minutes. So a total, the test is a total of 3 hours. They say 20 minutes on the first 2 and 6 minutes on the last 4. That's a lot of extra time then, isn't it? Oh, tomorrow's quiz is 20 multiple choice. You get 20 minutes. Okay, I guess that's it, huh? We went over the Google slide. Okay, so let me stop the recording here. And thank you for watching the YouTube video.